Hello and good morning, everyone. It's lovely to see you all today. Thank you for joining us from the comfort of your home. Welcome to the first Tuesday series with New England Biolabs, NEB, which is Amplifying Atcg U, presented by NextGen Scientific in partnership with NEB. My name is Fateen Ibrahim from NextGen. I'll be responsible for moderating today's webinar session and few more sessions to come in the following weeks. Before we get started, I'm going through some housekeeping to ensure that you can interact with us effectively during this webinar. Firstly, we would like to request everyone to put your mic on mute. If you wish to ask the speaker your questions, you may click on the raise hand icon on the bottom right screen, or you may use the Q&A activities feature by clicking the shapes icon and post your questions there. We also have an open chat feature enabled. To access this and chat with other online attendees, simply type and post there. Now, before we dive further into today's topic, I would like to give everyone a brief introduction of our principal company, New England Biolabs, or better known as NEB. NEB Company was established in 1974 by Donald Tung as a cooperative laboratory of experienced scientists. Initially, NEB produced restriction enzymes on a commercial scale. Its headquarters are in Ipswich, Massachusetts, America. The company has established subsidiaries around the world to maintain high-level service to customers, inclusive of Singapore, Canada, China, France, Germany, Japan, the UK, and Australia. While to the rest of the world, NEB maintained the service through distributors covering hundreds of other countries. And we here at NextGen Scientific are so proud to be appointed as the exclusive distributor of NEB to support the researchers here in Malaysia. NEB initially produces and supplies recombinant and native enzyme reagents for the life science research. In the recent years, NEB has expanded its product portfolio by providing solutions supporting nucleic acids isolation, genome editing, synthetic biology, and next-generation sequencing library prep reagents. NEB also provides a free access to research tools and scientific resources available through their website. Here, I would like to share with everyone a short video on the overview of NEB. Let's have a look.
We are involved in experiments and research which we believe is important, significant, and exciting. Ones which we are prepared and willing to share with other people, and happy to do so.
We have biochemists, molecular biologists, geneticists, all contributing to expanding the basic knowledge.
The goal of our company has always been to provide useful tools to the scientific community.
I can't say enough about how committed everybody is at New England Biolabs to serving our customers.
It helps breed that passion and that common sense of purpose and that family atmosphere that New England Biolabs has been known for.
Across the board, there's a culture here that actually takes full accountability for all the products that we produce and all of the activities and service that we provide to our customers.
There is a very collaborative attitude among the scientists here.
I love to see publications come out where scientists have used our products in something that's fun, new, exciting.
If it's got anybody's name on it, it has to be excellent.
Innovation is coming from our curiosity to understand a process and solve a problem, and we go toe-to-toe with anybody else doing science.
The focus is on science, and I think that sets us apart from other companies.
We have a lake here, we have a stream, we have woods. I wanted to protect the environment around here as much as we could.
We have an incredible environment around us, and coming to work every day is a real pleasure.
It's really just a great place to be.
I think people are just in awe sometimes of that it's actually here.
would now like to introduce our speaker today, Mr. Tung Chee Hong. Chee Hong is a field application manager at NextGen Scientific with the experience of more than 13 years. Chee Hong obtained his Master of Science degree in Molecular Biology from University Putra Malaysia UPM. He is experienced in molecular biology testing, inclusive of DNA-RNA isolation, PCR, qPCR, cloning, primer and probe designing, and also Western blotting. Chee Hong has also involved in cell biology testing of general cell culture and stem cell application, as well as diagnostic of diseases, HLA typing and cell isolation, microbiology testing, NGS, and CRISPR application. Not only that, Chee Hong has assisted customers with molecular diagnostics, such as screening for infectious diseases. Today's series is all about uncovering the most common DNA amplification method in molecular biology, which is polymerase chain reaction PCR. We will learn the tips and tricks of successful PCR and the PCR product ranges that NEB offers. With this, I would like to welcome Mr. Tung Chee Hong. Over to you, Mr. Chee Hong.
Hi, good morning, everyone. My name is Chee Hong. Thank you for joining me on the NEB Tuesday series today on the topic Amplifying Atcg U. Okay, so without further ado, let me get started. Okay, so currently there are a few DNA amplification technology available, and in NEB, we're actually offering reagent for a few types of DNA amplification. For example, we offer enzyme for loop-mediated isothermal amplification, which actually short form into LAMP, L-A-M-P. Okay, so besides, we also offer reagent for helicase-dependent amplification, that using helicase to open up the double-stranded DNA and then to perform the amplification. Last but not least, we are offering PCR reagent, which is polymerase chain reaction. So however, today we'll be focusing on polymerase chain reaction reagent only. Okay, so PCR or polymerase chain reaction is a well-known and widely used techniques for DNA amplification. Okay, so the power of PCR is that with just a few copies of starting material, we are able to generate million copies of DNA at the end of the PCR amplification. So in theory, PCR product number increase exponentially. So if you are referring to the diagram below here, okay, let's say we have a template DNA that started with just 2 copies of DNA here. So 1, 2. So at the end of the first cycle, we actually get the double number of the DNA copy, which is 1, 2, 3, and 4. And then followed by another cycle, which is the second cycle. We're able to generate 8 copies. And then next cycle, 16 copies, so on and so forth. A typical PCR reaction will consist of approximately 35 cycles, and in theory, we are able to generate approximately 68 billion copies of DNA. So since PCR invention in 1985, there are many applications that are already using PCR and its modified version. So here I have listed out some of the applications of PCR. Okay, so the first one will be gene cloning. So PCR will be responsible to amplify a full-length gene for gene cloning application, and then it will be used for recombinant protein production purpose. So in research, we also use PCR for gene expression study to try to understand the gene function and also gene regulation. Together with another technology that called sequencing, PCR and sequencing are able to perform genotyping analysis that allows the scientists to understand about variants and mutation happens to a particular gene. More recently, PCR also has been used for diagnostic purpose. So PCR has been used to detect presence of virus or bacteria from patient sample, that the doctor to actually to diagnose whether that the virus or bacteria is the causative agent of a disease. So in addition, PCR also has been used for phylogenetic study. So we generate PCR fingerprinting to study kinship analysis and also to get DNA fingerprinting. Last but not least, PCR has been used for genetic modified organism detection, GMO. So PCR has been used to detect presence of transgene in GMO. So to perform PCR, first we need DNA. Okay, so we need a sample with DNA. So the first step is to lyse the sample in order to extract the DNA. After getting the DNA, so for notes, okay, not the full DNA will be amplified in PCR. So PCR will only targeting a short segment of DNA and amplify the copies of this particular segment of DNA that actually defined by primers. So after getting a DNA, we actually mix together with all the essential components of PCR reagent together with DNA inside a tube. The tube will then transfer into the PCR machine. So the PCR machine will then generate many copies of the DNA. A basic PCR cycle consists of 3 steps. Okay, so these 3 steps we are referring it as a single cycle. So the 3 steps involved in this cycle actually are: number 1, denaturation that separates double-stranded DNA, followed by the step 2, annealing that allows primers to bind to a template DNA. And the third step, we have extension, in which DNA polymerase will incorporate nucleotide to the new elongated chain. So in order for denaturation of the template DNA to happen, we normally use high heat. So if you look at here, we have 94 degrees Celsius for 1 minute to allow separation of double-stranded DNA. After the finish of 1 minute, 94 degrees Celsius, we actually cool down the temperature to approximately 50 to 60 degrees Celsius for approximately 45 seconds. So at this temperature, the shorter primer are able to anneal or bind to the template DNA. So note that for PCR to happen, both forward and reverse primers is required to bind to the double-stranded DNA. Followed by annealing, we actually again increase the temperature to 72 degrees Celsius, which is actually the optimum temperature for Taq DNA polymerase to work. So the DNA polymerase at 72 degrees Celsius will start to elongate the chain initiated by the primer. Here showing a list of PCR components reagent that is required for PCR to work. So in addition to the template DNA here, okay, we need to have DNA polymerase. We need to have a buffer that maintain pH and optimum environment. We need magnesium chloride, which is actually a core factor for DNA polymerase. And of course, we need primers and the substrate, which is a DNTP. So each of these components need to be at an optimum concentration for PCR to work. A typical PCR reaction actually has a final volume of approximately a final volume of 25 microliter, or in another usual final volume will be 20 microliter. Once we have mixed all the reagent inside a tube, now we need to transfer it into a PCR thermal cycler. So in the PCR thermal cycler, we will see the standard cycling condition, denaturation, annealing, and extension. However, we have additional step, which is initial denaturation that actually incubate longer for at a high temperature. So first step, initial denaturation, followed by second step, which is denaturation, then annealing, and then finally extension. So at the end of the fourth step, we actually ask the machine to go back to step number 2 and repeat this particular cycling again. So 2, 3, 4, and then repeat again. So the repeat is actually 29 cycles, so which give a total of 30 cycles. When the cycling has been completed, then we'll proceed to the final expansion, and then after the whole reaction has been finished, then we hold at 25 degrees Celsius. So at the end of PCR, we're actually expecting a single bright and beautiful band smiling to us. Okay. However, sometimes this is not always the case, unfortunately. So therefore, we might require PCR troubleshooting. Some of the problem of PCR includes, but not limited to, number one, you don't see any desired band at all. Number two, you get bands in addition to bands that you want. So you have multiple bands. And third is you actually get contamination. So now we'll take a look at what kind of troubleshooting we have for this kind to deal with this kind of problem. Okay. So the first thing is, if for a new user of PCR, I would recommend to check on pipetting. So this is because PCR require good pipetting techniques and practice. Okay. So some of these good practice involve make sure that the pipette and the tips are actually fit tight. Okay. So make sure that the fitting of the pipette and tips is tightly. Okay. Secondly, after aspiration, check the tips to make sure that the liquid has been drawn into the tips. And after you dispense the liquid into the PCR tube, check again the tips, make sure that liquid has been really dispensed into the solution. Another tip is you may actually want to move your tube, as shown in this particular picture here, upwards after addition of liquid, indicate as an indication that liquid has been added. So the most crucial factors for successful PCR actually is the primers. Primers is a short oligo that normally size between 18 to 25 nucleotide. The sequence of the primer will affect the specificity of the reaction. Okay, so normally when we purchase the primers, it actually comes in lyophilized format. So we'll need to do the reconstitution, and generally we'll dilute the primer before use. An unsuccessful PCR reaction normally caused by poor primer design. Okay, so poor primer design can lead to primer dimer. As you can see in the picture here, there are some additional band below 100 base pair. So normally it's actually caused by primer dimer, and poor primer design can also lead to unspecific products. For troubleshooting of primers, we can perform a primer optimization using gradient PCR. Gradient PCR means that we actually run a series of different annealing temperature, as shown in the picture here. Okay, we try different temperature, let's say from 50 degrees Celsius, approximately 51, 52, 55, 58. So we try different annealing temperature to see which one actually is the best temperature for primer to work. So how do we decide? It's actually based on how bright and thick is the band, the desired band, and also whether there are unspecific bands generated in that temperature or not. So we select a temperature that gives the best U, brightness and density, and without any additional band. Another important factor for PCR to work will be the magnesium chloride. This is because Taq DNA polymerase is a magnesium-dependent enzyme. So optimum concentration of magnesium is required for the success of PCR reaction. So normally PCR, this magnesium chloride come in the format magnesium chloride or magnesium sulfate. So sometimes inadequate thawing of magnesium may lead to some weird PCR result. Another thing to note is that a lot of the commercial PCR buffer actually already contain magnesium chloride. So if, let's say, users are facing problem with faint band or no amplification, so one of the way is to actually increase the concentration of magnesium chloride. So higher magnesium concentration make the DNA polymerase easier to amplify. However, too high concentration of magnesium chloride also brings to another problem, because the DNA polymerase can amplify more easily it might also amplify unspecific bands. Next, we'll look at another factor that affects the successful of PCR, which is DNA. So if you have a failed PCR reaction, so one thing we need to check is whether you actually has enough input of DNA into the PCR reaction or not. The recommended amount of DNA that required for a PCR reaction is approximately 100 to 250 nanogram. So in order to determine the concentration of DNA, you can use microvolume spectrophotometer. You can also run the DNA, the extracted DNA on gel to see whether we can detect any genomic DNA or not. In another occasion, sometimes the failed reaction might be caused by inhibitors in the genomic DNA. So in this case, user might want to consider to perform dilution of DNA that reduce the amount of inhibitor in the assay, thereby increase the chances of successful. So sometimes you might observe that there are some smear DNA showing here. Okay, so these are DNA degradation. However, PCR is quite handle this kind of DNA degradation quite well. So normally it will not generate an issue for you. Another possibility that we generate a failed PCR is because of high GC content of your template DNA. So if you look at the targeted sequence of your PCR and you realize that it actually contains a high GC content, okay, it's most likely going to cause you problem for amplification. In this case, you might want to consider to add additives like bovine serum albumin or DMSO. So some of the commercial enzymes, for example, from NEB, one of our product, one tag, is actually come with a buffer that can work on high GC content. So we have a reaction buffer that is called GC reaction buffer that can actually work on template that with high GC content, for example, more than 65% of GC. In another product of NEB, we have Q5. So we actually supply with a high GC enhancer. So this additive supply with the kit will deal with the high GC content of your template. Okay, now we move on to another topic, which is to know the crucial component of your PCR reaction, which is the enzyme. So the question is, do you know that there are DNA polymerase other than TAG? And why do you need this enzyme compared to TAG? Okay. So TAG DNA polymerase was first isolated from a bacteria named Thermus aquaticus from a hot spring here in Yellowstone. The unique features of TAG DNA polymerase is that it is able to withstand high temperature, which is good for PCR because we have the denaturation step. After the discovery of TAG DNA polymerase, scientists have been trying to improve the performance of TAG by modifying the DNA polymerase. In addition to that, we also try to isolate some other DNA polymerase from other sources. So from NEB, we have a DNA polymerase that named as 1-TAG. 1-TAG is actually an optimized blend, means mixture of TAG DNA polymerase and deep van DNA polymerase. So deep van is actually a DNA polymerase isolated from another bacteria named Pyrococcus. This organism was isolated from a submarine thermal vent that is actually 2,000 meters below. Okay. So when you're using modified enzyme, so be sure that you need to check on the product sheet because some of these modified enzyme might not work the same way as TAG DNA polymerase. For example, from the slide earlier, I mentioned that 1-TAG from NEB, the optimum extension temperature is actually 68 as compared to 72 degrees Celsius of this TAG DNA polymerase. And for Q5 DNA polymerase, if you look at the denaturation step, okay, over here, the recommended denaturation step for Q5 is actually 98 degrees Celsius. Also, again, for Q5, the primer concentration is higher. We recommend to use 0.5 micromolar instead of 0.2 that usually use. And also for annealing temperature, Q5, we actually recommended melting temperature +3. Okay? For normal DNA polymerase, we actually recommended melting temperature -5. Okay. So now you might be wondering, why do you need modified enzyme? Why do you need something other than Taq DNA polymerase? Okay, so the simple answer is because not DNA polymerase are created equal. Okay. So what I mean by that is, let's say for a special application, we have here multiplex PCR. So in multiplex PCR, we actually add several pair of primers inside a single PCR reaction. Because there are many primers in the PCR reaction, they might actually interact and produce unspecific products during PCR setup. This means that this thing happened before the actual PCR event happened. Okay? So to tackle this unspecific amplification during setup, the scientists added a function called hot start to the polymerase to prevent unspecific amplification. A typical hot start function will be shown here. The scientists are actually using antibody to bind to the active site of the DNA polymerase. Therefore, during setup or room temperature, the enzyme is actually inactive. Okay. However, when we start the PCR reaction, so the first step is actually denaturation, a very high temperature. Okay. This high temperature will actually denature the antibody and therefore free the active site of the enzyme. So after we initiated or started the PCR, the enzyme actually become active and ready to amplify. So in NEB, we actually go one step further. So instead of using antibody, we are using aptamer as the hot start blocking. Okay. So why does it matter? Okay, if you look at here, NEB, when we're using aptamer, the advantage is that we do not need any activation step. So when compared to those using, let's say, antibody, they might need 15 minutes incubation, 1 minute, 4 minutes, and they need some incubation time. So therefore, by using aptamer, we actually save some time, okay, for the PCR reaction. So it will finish faster. Okay, so another occasion where we might actually need a modified enzyme is because we are trying to PCR a full-length gene. Okay, so for a PCR of a full-length gene, I will actually recommend the use of a high-fidelity proofreading DNA polymerase. So what is this high-fidelity proofreading? So one of the applications of using this is, of course, PCR full-length gene for cloning purpose. So the specialty of this particular high-fidelity proofreading tag is that it has low error rate. Okay? So low error rate means that it's actually highly accurate in terms of the complementary base pairing. So they make very little error, and most of the time they actually added the correct base. Another features of this enzyme is that it's able to perform self-correction. So in the event that they actually add the non-complementary base into the extension, they will actually perform a double check. Once they realize that it's the incorrect base pairing, they will actually use the exonuclease activity to remove the wrong base, and then again they added another base again. So they have the self-correct function. Okay, so the NEB-Q5 is actually the polymerase with high fidelity proofreading activity. So as shown in this graph here, it's actually 280 times more accurate than a normal Taq DNA polymerase. So you might be thinking, I'm only amplifying a 2000 base pair gene. Okay. And if you look at here, a Taq DNA polymerase normally will only make one error per 3,300 bases. So I'm using 2,000. So it shouldn't pose any problem, right? Okay. So however, when we look at the PCR amplification, we know that the same 2000 base product will be amplified over and over and over many times. Therefore, we are not talking about 2000, but we're talking about more than billions number of base. So therefore, we need to have high accuracy to make sure that our product is accurate. So in a PCR fidelity estimator from NEB side, you can see that with a 2000 base pair PCR product, okay, so after 30 cycle of PCR, Q5 will have 98.8% correct copies. Means that 98.8% of them will actually, the sequence is accurate. Okay, whereas for tag, only 4.7% of those amplicons has correct sequence. So this is quite significant. Okay. So if you're looking for a faster PCR, it's also possible, but mostly we are talking about a long amplicon. So NEB-Q5 DNA polymerase has been modified to have the SSO70 binding domain. So with this modification, Q5 is able to amplify faster, and then thereby shorter the amplification time. Okay, so if you look at here, Q5 actually has a 10 second per kb amplification. Means that if you have a 5 kb amplicon, you only need 50 seconds for amplification. Else for a standard tag DNA polymerase, you might need like a few minutes for the amplification. Okay. In addition, because of this binding domain, Q5 actually has improved affinity, meaning that it's actually more stable binding to the template. Therefore, it has increased amplicon length. So for NEB-Q5, we're able to amplify amplicon as long as 20 kb for simple amplicon and 10 kb for complex amplicon. So whereas for tag DNA polymerase, the normal length is only approximately 5 kb. Okay, so if you would like to further reduce the PCR runtime, you might actually using, let's say, master mix. So master mix is actually a pre-mix that added all the essential components except primers, template inside. So therefore, you have fewer pipetting steps. So make your whole setup faster. Another thing is you might use a buffer that with loading die. Okay, so with color. So therefore, at the end of PCR reaction, you can straight away load the PCR products into the gel. Okay, you don't need to spend time mixing with loading die anymore. And finally, if you have more budget, you might want to consider using a thermal cycler with silver block. Okay, because the silver is actually a better conductor. So therefore, the whole PCR process, the 3-step cycle, will actually happen faster. Okay, so thereby shortening the time required for the PCR to complete. Okay, so in another occasion, for a special application, you might be dealing with modified base. So in this occasion, you might be amplifying U, yeah, referring to DUTP. So we need to select a special polymerase that can actually recognize modified base. So whenever we deal with template that with uracil or inosine, we might need this polymerase. Okay. The application that actually has a template of U or inosine are, okay. So when we are dealing with methylation PCR, okay, the converted DNA actually contains UTP. Okay. And in another occasion, if you're using primer with inosine, okay, so you cause the template to contain inosine as well. Okay. In these two occasions, we need a polymerase. Okay, a polymerase showing here that can actually recognize the DUTP or inosine. So one of the choices will be Q5U, okay, which is a special version of Q5, or EpiMart hot start tag. Okay, so before I finish my presentation, I would like to share to you an experimental setup of PCR. Okay. So normally for experiment, we will need some validation. So we need controls to validate that our result is really positive, the positive is really positive and negative is really negative, okay, to validate the result. So some of these control in PCR include positive control. Okay, so positive control is used as a reagent and instrument control because positive control should always be giving you a band. Okay, so if something happened to your positive control means that no amplification, then a negative sample is not a true negative. Okay, something might be happening to your reagent or instrument, so therefore we need to check. Another control that we require for PCR will be no template control, meaning that no DNA in that particular tube. Okay, so in this tube, we are not supposed to have any band or amplification. In case we saw bands in no template control, it's actually a contamination and make the whole PCR reaction not valid. Okay, so because if the NTC is positive, it might give false positive result to your sample. Okay, so how do we avoid contamination and how we want to deal with PCR contamination? So the first recommendation is to use filter tips because a lot of time this contamination is actually template DNA or PCR product from coming from aerosol that contaminate your pipette. Okay, so the secondly, you might want to separate your reagent setup area when you set up your PCR mix. Okay, you want to separate from the place where you run PCR or you do gel electrophoresis. Okay, and good practice, another good practice will be use dedicated pipette set for reagent setup and gel electrophoresis. Do not mix them. And if you have the laminar hood, you might want to set up your PCR in the laminar hood to protect your sample and not to be contaminated by the surrounding that actually maybe contains the contaminant. Okay. And finally, if really required, you can use an enzyme called UNGase to destroy the PCR products coming from previous round. So somehow, if, let's say you spill a PCR product and then it contaminated the area, okay, so this actually becomes the source of the contaminant. So you might want to use the enzyme to destroy all these PCR products. Okay, so to sum up the sharing on today, okay, so PCR amplification generate many copies of the same DNA from small amount of starting DNA. Secondly, PCR result can be influenced by good primer design, DNA quality, and also magnesium concentration. Okay. Thirdly, enzyme other than Taq DNA polymerase may be more useful for multiplex PCR. It's good for high fidelity amplification for gene, okay, and also might reduce the PCR time that gives you a faster PCR. Another thing is when we are dealing with DUTP, we might actually need a special polymerase to amplify the U base. And finally, we can avoid cross-contamination using filter tips and good PCR practice. So with that, I shall end my sharing on today. Hope that everyone is getting some useful information or tips on today's sharing. Okay, so yeah, now I'll pass the floor to Fateen.
Thank you very much, Mr. Chee Hong, for the thorough and informative presentation, like the various details that we need to use to ensure successful PCR and also different types of DNA polymerases to use for specific PCR amplification. So now we will open the floor for questions. Let me check. Right. So do you have any questions so far?
Hello, hi, I'm Nisha here.
Hi, Nisha.
Yeah, okay. I just have two questions. I'm actually a parasitologist. I work with fecal samples.
Okay.
So I do collect my fecal sample from the Orang Asli village for parasite screening in alcohol. Okay.
Okay.
So 70% alcohol. So whenever I extract my DNA, I use a commercial kit.
Okay.
The quantity is good. Quantity is good and the purity is okay. But when we run the gel, it looks, it shows some degradation smear.
Okay.
Okay. And some, okay, not all, some are showing degradation. And that's my, I don't understand what's the reason. First, my second question is, I've tried using Tech Polymerase and PFU enzyme to screen for my worm. Mainly, I use PFU enzyme because I'm doing sequencing also after my DNA, after my PCR. But few instances, I found that whenever I use PFU, at times I don't get results. And when I change to either Tech Polymerase or hot screen, I get results. Why is this so? Thank you.
Okay, so thank you, Nisha, for your question. So the first question is about the sample. Okay, so to me, okay, the most important thing is whether you get result or not. So most of the time for PCR, right, they can actually handle degraded DNA quite well. So that's why if you plan to just PCR your extracted DNA, so no worries. Because shorter fragment PCR, normally we actually targeted shorter fragment. Therefore, we might still able to amplify most of the time. Therefore, we actually have a lot of occasion where we try to amplify PCR, amplify ancient DNA, or actually samples that are heavily degraded. Okay. So as for the reason why your sample actually contains so many degradation, so one of the possibilities is that I'm not sure whether you extract the sample on the spot or not, because sometimes when we actually doing transport, okay, so the sample might be already start degrading. So in that case, if your sample already start degrading, okay, so the DNA might also expose to the nuclease and also the environmental nuclease. So they might be already degraded before you actually manage to extract the DNA. So therefore, sometimes we actually have some transport protection for the sample. Okay. Another thing is that maybe the sample actually contains high amount of nuclease. Okay. So when you process the sample, you lyse the sample, the protection is not enough to actually protect the DNA from the nuclease, and therefore at the end of the extraction, you get degraded DNA. Therefore, I would like to highlight 2 points. One is sample protection, okay? So before you perform PCR. And second thing is your sample actually contains high level of nucleases? Okay. Okay. So and then as for your question about the PFU, okay, one of the suggestions is I would like to suggest is you can actually check with the support from where you actually purchase the enzyme, because I might not be knowing the enzyme very well, so it's best for them to actually recommend some optimization to actually make your PCR more successful. Okay, because as far as I understand, okay, sometimes PFU depends on which version you're using. It might be a bit choosy in terms of amplification. Okay, meaning that PFU sometimes has problem generating a thick band or bright band. So meaning that the amplification efficiency is actually slightly lower than TAC-DNA polymerase. Okay, so therefore when you are using TAC-DNA polymerase, okay, the amplification is okay because the TAC-DNA polymerase is quite robust. However, when it comes to PFU, depends on the version you use, it might not have the same amplification efficiency. Therefore, it didn't give you the bright band and nice band like the TAC-DNA polymerase. Okay, so I hope that this shall answer your question.
Yes, yes, thank you.
Right, thank you, Nisha. And then we have requests from Hassan and Nurul Aimi on the presentation. Yes, of course, we can share the slides and as well the recording of this presentation to everyone who participated today. So don't worry. And then the next question from Hassan Yahya: what is the maximum length of DNA a PCR can detect? So.
Okay, so if you are talking about the maximum length of genomic DNA, that is very long, Okay, but let's say, for example, a chromosome of a bacteria normally has around 3 million bases. Okay, so however, normally we will not amplify the full length of the 3 million bases. Another thing is when we actually perform the extraction, a lot of time because we are doing a lot of mixing and all that thing, okay, so the DNA might break into shorter fragments. So a lot of times, that's why the genomic DNA that we get, the size is approximately 20 kb, okay, 20,000 base pair. Although there are special kits that can actually extract near to full-length DNA. Okay, so as for PCR, okay, how long can we amplify? Okay, so that one will actually depends on the DNA polymerase that you are using. Okay, so as I showed earlier, different DNA polymerase has different maximum length of amplicon. So for example, for just now I mentioned that for Q5, you can actually amplify 10 kb, means 10,000 base pair for complex DNA. Complex DNA normally referring to genomic DNA. Okay, so for not so complex DNA, for example, plasmid, you can actually amplify approximately 20,000 base pair. Okay, so therefore you have to look for the specific enzyme that you're talking about. If let's say you have an application that need to amplify very long DNA, 20 to 30 kb, you can try to send your request to the salesperson and asking that, stated that you actually try to amplify PCR product that is, let's say, 20 kb. Ask them to recommend and show info about the tech polymerase, DNA polymerase. They are capable of doing so. Okay, so as far as I know, there is DNA polymerase claim to be able to amplify 30 kb. Okay, so if not mistaken, that is the longest I have ever seen.
Right. Okay. So next question is from Yuan Ting Hoi. For sequencing using primer walking techniques, we usually design many primers to send to a company with the long PCR products to get the sequences. So how many primers are considered sufficient or optimal?
Okay, thank you, Yuan Ting, for your question. So unfortunately, I'm unable to give you accurate answer as well, because this one is a bit depends on your template as well. Okay, so for primer working, so depends on the first primer, how long you are able to sequence. So ideally, okay, so if for estimation purpose, okay, so generally a standard sequencing should be able to give you information of approximately 800 to 1,000 bases. Okay, so we can actually, if you want to do, let's say, the standard sequencing, so we are expecting 800 to 1,000. However, more often than not, because the template DNA is a bit difficult, a lot of time we are talking about the difficult template. Okay, so that's why I will assume the size will be cut short to approximately 500. So therefore, if you want to design, so you can try the first one. Hopefully, you can actually reach 500, and then from there you continue. Then you expect it to be approximately 500, and from there again another 500. So for me, if I'm doing the primer working, my design will be expecting the sequencing length of each primer to be approximately 500.
Okay. Next question is from Alif Ihsan. I'm currently dealing with plant samples, specifically from leaves. I'm using the common DNA extraction techniques for plants, which is CT-AB method. My question is, do you have any suggestion to optimize the bands produced, maybe during extraction or PCR?
Hi, Alif. I'm not sure how can I give you a good answer? But to me, CTAP, as far as I know from all the customer that I actually meet before, is actually quite a good method for plant. Okay, so but maybe the thing is, I'm not sure whether you have included the liquid nitrogen grinding step. So even though CTAP can wash away a lot of the contaminant and non-purity from the plant sample, we might need to add in, let's say, a harsh method like liquid nitrogen to actually destroy the a very hard sample like this leaf. Okay, so I'm not sure whether you're already using that. Okay, so as for the band produced, do you mean that you get multiple bands? Okay, if you're getting multiple bands, more than one band that you're expected, then maybe you want to consider, I'm not sure whether your primer is actually obtaining from journal or you design it yourself. Okay, so if for optimization, you can try using the gradient PCR method, try different annealing temperature, and then see which one actually give you the best band. Okay, what else I can give you some suggestion. Yeah, I think, yeah, for optimizing band. Okay, so if let's say if this didn't work out, my another suggestion is if you're designing the primer yourself, maybe you want to consider design it again, because sometimes optimization, if the first time not working out, you might actually spend more time trying to optimize it. In that case, I will suggest you to actually make like a cutoff point. So after certain optimization, you might want to consider using a new pair of primers.
Okay. Next question is from FO. Number one, what is the cost of the smear apart from DNA degradation? I always obtain smear bands from my PCR reaction. The sample was actually a synthesized gene block. I tried running the original sample and it was okay. The purity was also okay. And number two, how to increase the efficiency for colony PCR? I did not get the desired band sometimes, but I managed to get the band when I ran the PCR using the purified plasmid. Okay.
Okay, so FO, the cost of the smear is actually a very difficult question because I never see your gel image, and I also not really sure about the handling of the product. Okay, so I am a bit unsure why you're getting smear bands from your PCR. Oh, so you're saying that you get smear from your PCR. Okay, so sometimes the smear can be caused by the plasmid DNA that, sorry, your template DNA. Okay, so and also sometimes, most of the time, the smear can be caused by your primers. So depending on your primer design, if you have unspecific amplification, you might see smear. Another thing is if your band is really concentrated, when you actually run gel, sometimes you observe a bit of a draggy as well. So I'm not sure is that the type of smearing that you're talking about. Okay, so maybe you can send your gel image to us, and then we take a look to give you some suggestion. Okay, so the second thing is how to increase the efficiency for colony PCR. I did not get the desired band sometimes, but I managed to get the band when I run the PCR using purified plasmid. Okay, so for colony PCR, I'm not sure whether you are using commercial kit. Okay, so or you're talking about conventional cloning. Okay, so for conventional cloning is a bit all depends on the ligation ligation step that you're working with. So try to increase the ligation efficiency. But if you're talking about the commercial kit, you might actually want to take note on, let's say, there are some suggestions on the ratio of the insert and the vector, and then you might want to do the molarity ratio calculation and all that. So it might help. Another thing is if you're talking about conventional PCR, so sorry, conventional cloning, then you might want to make sure that your restriction reaction actually perform completely, so that they have actually pairing ends. So again, I do not have enough information on this, so that you can actually increase your efficiency for colony PCR. But basically, is I think you're talking about clone with the correct sequence. So to me, the main thing is the ligation part. So try to increase the efficiency at the ligation part.
Okay. So we'll take the final question from Hasna Ahmad, which is, we routinely perform colony PCR direct from yeast colonies. What polymerase would you recommend for this application?
Okay, so for yeast colony, depending which yeast that we are talking about, so I think you're using the heat lysis step. So the important thing here will be the whether the polymerase can withstand inhibitors, because when we are using lyse yeast cell or bacterial cell, it might contains the inhibitors because we are not purifying it. Okay, so therefore you're trying to look for polymerase that can withstand inhibitors. From NEB, we will be recommending one-tag DNA polymerase. One-tag we actually can perform colony PCR, and for other brand, so again you can check with them whether their DNA polymerase is actually have good performance for colony PCR or not. So to deal with those inhibitors.
Okay, so thank you again, everyone, for the questions. That was very great interactions we have. Thank you again, Mr. Chee Hong, for answering the questions. So if you have any more questions, or would you like to, discover more product ranges that NEB offers, you can visit https://international.neb.com, or you can direct your questions to sales@nextgene.my. It was an absolute pleasure to have everyone with us today. Thank you all for attending. We hope to see you again next Tuesday at the same time, 10 a.m. for the series Lighting the ATCG for Quantitative PCR Method. Till we see you again next week, thank you all and have a good day ahead, everyone. Thank you.
Hi, thank you, everyone. Please join us.
Thank you.
Thank you.
Please join us again next week.
Thank you so much.
Thank you.